Today I will be explaining urea cycle. So let's begin the presentation. So learning objectives. What will you learn about in this slide? So firstly you learn about all the steps involved in the urea cycle and the enzymatic activity in the cycle. So what sort of steps are involved in the urea cycle and what sort of enzymes are responsible for carrying out the entire urea cycle. Okay, now what is urea cycle? So urea cycle can be defined as series of biochemical reactions that convert toxic ammonia into less toxic form that is called urea. So basically this is ammonia compound and this is urea. So we carry out this cycle because we want to convert this ammonia, which is a very toxic compound, into less toxic form that is called urea. And where does this ammonia comes from? Basically this ammonia comes from the degradation of amino acids in the body. As variety of proteins are being produced in our body, and whenever these proteins are catabolized, they're going to produce amino acids. And these amino acids are degraded and they'll produce variety of nitrogenous containing compounds, and some of them are very toxic like ammonia. So this ammonia is a very toxic compound which cannot be absorbed into the water. So what we have to do, like we have to convert this more toxic compound, ammonia, into less toxic form urea so that it can be excreted out of our body through urine. Now the site of urea cycle. Where is urea cycle occurring? So basically urea cycle, it occurs in the mitochondria and the cytosol of the liver cells hepatocytes. So you can see in this diagram that this is liver and here the urea cycle is occurring. First two steps of the urea cycle, they occur in the mitochondria of the liver cells and remaining occur in the cytosol of the liver cells because the enzymes responsible for catalyzing the first two steps, they are present in mitochondria, while the enzymes responsible for catalyzing the other steps, they all are present in the cytosol of the liver cells. And you can see in this diagram, after the formation of urea, it gets into the bloodstream and then it is going to pass through the tubules of the nephron in the kidney, and the excess amount of urea, it gets excreted through the body in the form of urine. Now the reactions of the urea cycle. So the first reaction is the formation of a compound named as carbamyl phosphate. Okay, and then we have got the formation of citrulline, and then there is formation of arginosuccinate. So there are three steps: formation of carbamyl phosphate, formation of citrulline, and synthesis of arginosuccinate. The first two steps, they're occurring in the mitochondria of the liver cells because their enzymes are present in the mitochondria, while synthesis of arginosuccinate, it occurs in the cytosol of the liver cells. Then we have got cleavage of arginosuccinate, cleavage of arginine to ornithine and urea. Finally, we have got urea here. And then we'll discuss a little bit about the fate of urea, like what is going to happen to this urea, what is going to happen after we have successfully conducted a urea cycle. So what is going to be the final destination of this urea? You can see this is a urea compound and it has got two nitrogen atoms and a doubly bonded carbon atom as well. Now, the first step of the urea cycle, that is the formation of carbamyl phosphate. You can see this mitochondria written here because this reaction is occurring in the mitochondria of the liver cells. So, what are the reactants of this reaction? So, we have got a toxic urea, a toxic ammonia compound, then we have got carbon dioxide. They both are going to combine together to form carbamyl phosphate. And this reaction is an ATP dependent reaction that requires two molecules of ATP. And the most important enzyme that is responsible for conducting this reaction is called carbamyl phosphate synthetase that is present in mitochondria. Apart from that, we have got an activator that is called N-acetyl glutamate that is going to facilitate this reaction. Next step is the formation of citrulline compound. So we have got carbamyl phosphate that was already synthesized by the combination of carbon dioxide with ammonia, and this carbamyl phosphate is going to combine with the precursor ornithine that is already present in mitochondria to form citrulline in the presence of an enzyme called ornithine transcarbamylase enzyme, which is present in the mitochondria of the liver cells. After that, we have got synthesis of arginosuccinate and then cleavage of this arginosuccinate. So citrulline that was already produced, it is going to combine with aspartate and then it is going to form arginosuccinate. And in the next step, we'll see that this arginosuccinate is going to get cleaved to produce two products. So this reaction is basically occurring in the cytosol of the liver cells because its enzyme is present in the cytosol, that is argininosuccinate synthetase. Now we have got the cleavage of arginine to ornithine and urea. So this was argininosuccinate and it is cleaved into fumarate. It is cleaved into fumarate. and this fumarate is going to combine with it to form arginine, and this arginine is going to break down into urea as well as the precursor ornithine. So this is the urea that we wanted, and it is a less toxic compound, water soluble. It can easily travel into the bloodstream, move towards the kidney where it gets reabsorbed, and excess amount of urea it gets excreted into the urine. So now this is an overall summary of all the reactions that we've studied so far. So first of all, we've got this carbon dioxide, and then we have got this ammonia. They both are going to combine in the presence of carbamyl phosphate synthetase enzyme that is present in the mitochondria of the hepatocytes, and it is going to produce carbamyl phosphate. This carbamyl phosphate is going to enter the urea cycle and combine with ornithine that is already present in the mitochondria of the liver cells to produce citrulline, and this reaction takes place in the presence of an enzyme called ornithine carbamylase transferase. After that, this citrulline is going to combine with aspartate to produce arginosuccinate. This reaction is occurring in the cytosol of the liver cells in the presence of argininosuccinate synthetase enzyme. This argininosuccinate is going to combine with fumarate to produce arginine in the presence of lyases, and this arginine is now going to get cleaved into urea as well as ornithine. When it combines with oxygen, it forms ornithine. When it combines with water, it forms urea. So this is the urea that we wanted, and this is the precursor ornithine that is already synthesized, and it is going to continue the urea cycle again. So this process will continue on. And finally, we'll look about the stoichiometry of the urea cycle. So what are the reactants and the products that are produced in the entire cycle? So first of all, our reactant was ammonia that was combining with carbon dioxide. Here, the source of carbon dioxide is bicarbonate ions, and it is going to produce urea. And then we have got aspartate also, which also combines with certain products to produce fumarate. And then we have got three ATPs because they were utilized and converted into two ADPs. Along with that, we have got adenosine monophosphate, some inorganic phosphates, and water. So this was the entire urea cycle, and this urea cycle is basically conducted to convert to convert more toxic ammonia into less toxic urea. That was all from my side. Thank you so much.